Hi, my name is Hugues Crollius, and it's a pleasure today to present ATLASea, a newly funded biodiversity genomics project that aims at sequencing several thousand genomes along the French coastline. So let me first address the question of why in France and why now. So as you know, there's a convergence of several projects under the umbrella of the Earth Biogenome projects that have started recently. One of them is the Darwin Tree of Life, fairly close to us. And of course, there's ERGA, the European Reference Genome Atlas. So to start this project, it seemed important to focus on the marine ecosystem because France has a very large maritime zone, the second in the world after the United States, and approximately 45% of the French coastline is protected. And one reason for this is because marine ecosystems are fragile and under high anthropogenic stress. And so it seemed urgent to document this biodiversity, to understand it, and to better protect it. So another reason is that marine species are relatively unexplored and unknown. So it's also important to focus on this biotope. So globally, ATLASea's objectives are to sequence approximately 5,000 genomes along the French metropolitan coastline, and then another 1,000 genomes along several island shores in the Pacific Ocean, Indian Ocean, and Caribbean. So altogether, to generate massive amounts of biological data of high intrinsic value, stimulate and structure the French community through 2 pilot projects, and of course by providing a lot of genomic data, and also connect with the economic players for innovation. So why 6,000 species? Well, the French Biodiversity Office has already documented over the last decades approximately 12,000 species along the French metropolitan coastline. And so we thought that starting with a first aim of 6,000 would cover a broad range of taxonomic groups and allow us to also explore new species. You see, nevertheless, that a lot of these taxonomic groups are colored green and red, which, as the legend shows, suggests that at least 50% of that they only represent about 50% of what is potentially known. And so we know that these are vast underestimates in terms of known taxonomic groups, but well, you have to start somewhere. So the roadmap for ATLASea is to sample along the project in several marine stations, but also organize sampling expeditions, both in metropolitan France and in islands overseas, prepare high-quality DNA, sequence, assemble, establish an informatics infrastructure for storing, sharing, and displaying the data, and we have also teaching, training components in the project, and then 2 pilot projects that aim at exploiting the data in a way that demonstrates how genomic data can be useful across the range of biological research. And then we also will have an innovation call to enable industrial partners to use the data as well. So the governing structure of ATLASea is such that there are 7 institutional entities that will steer the project, CA and CNRS, 3 universities, Sorbonne University, Aix-Marseille University, and Paris Sciences Lettres, and then the Natural History Museum in Paris and Ifremer. And then there are several research infrastructures and organizations that will be part of the executive committee. As you see, there's ERGA, represented by Rob Waterhouse. There's the Terra Océan project. There's the European Marine Bioresource Center, the French Bioinformatics Institute, and so on. So the general project is organized as one large pipeline where we will have sampling expeditions. DNA will be prepared at Genoscope, the French Sequencing Center, for high molecular weight DNA, and also for DNA for short read and high C. And then the genomes will be assembled, annotated, and then shared across public repositories and internal databases, and will be accessed by the players in the pilot projects. So the sampling expeditions will be focused on marine stations, and so in Villereux in the English Channel, in Roscoff in Brittany. Also, we will take a boat to the Rochebonne Plateau, which is off the Atlantic coastline, where there is a high-rise plateau which is rich in biodiversity. There are 3 spots in the Mediterranean, in the Banyuls Marine Station, the Andoum Station, and Villefranche Marine Station, and then 1 expedition at the south end of the Corsican island. And then 4 spots as well in Martinique, 1 in the Caribbean, in Mayotte in the Indian Ocean, and 2 in the Pacific Island, in New Caledonia and in French Polynesia. There are also a lot of collections available in marine stations that we will be sampling, and are backed by strong biological interest. And then the pilot projects target the 2 ends of the spectrum of biological research. The first one is really focused on molecules, so molecules of interest generated and produced by animals and plants and algae in the marine ecosystem that are already of interest for agriculture, for medicine, for industry in general. And so the first component of this pilot project will be essentially computational, with a strong effort at functionally annotating all the genes and proteins from the 6,000 genomes, putting them into an evolutionary context in phylogenetic gene trees for comparative genomics, and then we'll be targeting pathways leading to these molecules of interest, which are already known, the molecules, but the pathways are unknown. And then we'll do comparative genomics to take to leverage the availability of the multiple genomes for taxonomic groups to look for variations in the pathways that lead to molecules of different activities, different substrates, and so on. And then there is an experimental phase that will take model organisms and then characterize these pathways in the model organisms in microalgae, macroalgae, and animals. The second project is at the other end of the spectrum, is going to look at the ecology of marine species in the context of invasions. So this has to do with generally understanding biodiversity, how it evolves in a certain ecosystem over time, but also for biosecurity reasons, economical reasons, and so on. So the question is, how do invasive species become successful in a given environment? What happens at the genomic level? Are they hybridizations? Are they polyploidy? We will study genetic diversity by resampling individuals in the wild based on the reference genomes to model the demography and potentially discover key genomic features that are associated with successful invasions. So I'd like to finish by emphasizing the fact that ATLASea is set in a favorable context for running such a project at the national and international level, with, for example, of course, the emergence of ERGA, the European Reference Genome Atlas, the backdrop of Elixir, and the synchronicity with the UN Decade of the Ocean program, for which I think we share the same motto: the science we need for the ocean we want. So thank you very much for your attention. Goodbye.